So welcome back, everyone. This is the afternoon session of the first inaugural Canadian Computational Neuroscience Spotlight. So this morning we had microscale oscillations. This afternoon we're moving up a level and we're going to be looking at macroscale oscillations. So we've got 3 talks for you this afternoon. I'll be giving a tutorial, relatively short, next 20 minutes, talking about neural population models for macroscale brain oscillations. Then we have a research talk from Jeremy Lefebvre, who's going to be talking about large-scale oscillatory dynamics and white matter. And our keynote this afternoon is Dimitris Panoutzis, who will be talking to us about predictive coding in cortical microcircuits and stuff like that. So without further ado, I'm going to crack on with the first talk of the afternoon, which is my own. I'm going to be judge, jury, and executioner today because I'm giving the talk and moderating the panel discussion for this session. Great. So before I get into the nuts and bolts, I feel like a few words are in order on terminology, which is that some of the words that we're using here, some of the words that I'll be using, some of the words that are even in the the conference title and the session titles, they're somewhat not particularly concretely defined. And really, I'm talking about mesoscale and macroscale. So to the uninitiated, this is a schematic of some of the scales and some of the types of structures that we're often studying in neuroscience, going from the small scale to the large scale. On the left-hand side, we have starting with neurons, and the definitions there are fairly concrete. It's in the name, microscale, talking about structures that are on the order of microns. Now, typically, textbooks and review articles and so on, mesoscale in neuroscience is generally given as structures and systems on the order of millimeters, and macroscale structures and systems on the order of, let's say, centimeters to tens of centimeters. So, that's a kind of rough numbers that you'll often see, but it's important to know that there's a lot of fuzziness in the middle. And in a nutshell, like one person's meso is another person's macro, and one person's meso is sometimes another person's micro. So it's just important to be aware of this when you're listening to talks and consuming the literature and so on, that there's a lot of fuzziness in the middle. What I'm going to be talking about here is neural population models. So you see in my schematic here, we have neural populations down near the bottom left. So the idea here with neural population models is to find a quantitative mathematical description that we can use to describe the average or the collective activity of large numbers of individual cells. So we want to basically have a low-dimensional set of equations that describe everything that's going on in this diagram here, like these thousands or hundreds of thousands of cells that you see in even a small, couple of cubic millimeter slice of tissue. Why do we want to do that, or why do we think that's a good idea? So that's a whole talk in itself. But one of the best answers to that question, to my mind, is the fact that we can measure structured macroscale neural activity with tools like EEG and MEG. fMRI going to count here as well. So essentially, like if there wasn't meaningful population average collective activity happening in the brain, then we wouldn't be able to see anything with EEG and MEG. So it kind of serves as an existence proof for the presence and the motivation of aiming for descriptions at that spatial scale. And then beyond that, you want to be aiming when you're developing models and trying to, build knowledge. You want to be aiming to have some commensurability between the spatial scales of your measurement and the spatial scales and the level of detail of the models that you're using. So that's the type of motivation that we think of when we're using neural population models to describe signals like EEG and MEG. So the types of models that are used in the field, I have just a few listed here. This is a non-exhaustive list. There's lots missing. Please don't shoot me if your favorite model is missing from the list. One of the things that you'll often encounter is this difference, this kind of categorization of biophysical or biophysically motivated and phenomenological or phenomenologically motivated. Really is more of a, I would say, like a continuum rather than a binary classification, as in some of the, say, the models on the left-hand side, for example, and others you might put in that list, are have more or less levels of physiological and biophysical detail, and some of your phenomenological models might have more or less levels of abstraction. But nevertheless, this is a kind of categorization that you'll often see, and in the talks that you'll hear this afternoon, there'll be the representation from both of these. Jeremy will be talking about Kuramoto models, and Dimitris will be talking about Janssen-Rit type systems. And for this tutorial, I'm also going to be focusing on the category on the left-hand side. So I'm going to give you an overview of some of the key architectural and conceptual components of it's like a canonical type of neural mass model. And I want to go in that through a brief historical segue, which is via the schema that was proposed by a scientist named Walter Freeman, actually in the 1960s, and really developed in the 1970s, called the K sets. K stands for a chap named Katchalsky, who these are named after. So you can see I have my, my spatial scales of neural structure slide just up here in the right-hand corner. The idea with the K sets is it's a framing. It actually has a full kind of specified mathematical formulation, which I won't go into much here, but it's a framing of a way of thinking about nested hierarchical levels of structure in neural systems, and particularly at the meso to macro spatial scale. So what we have is at the base level, what's called the K0 set. And the K0 set is a isolated, if you like, neural population model that is of a single type. So all excitatory or all inhibitory neurons, all with globally common inputs that are uniform, if you like. So moving up a level, we have the K1 set, which is pairs of interacting K0 sets, but are still all uniformly excitatory or inhibitory. And then the next level is K2, and K2 is the most interesting and most kind of enduring aspect of component of this schema, which we'll come back to. But K2 consists of an excitatory K1 set interacting with an inhibitory K1 set. Okay, and then to finish this off, we have K3, which is multiple interacting K2, and then K4, multiple interacting K3. Now, I don't have time here, and this isn't the place to go into the various systems that this has been studied and the pros and cons of this full schema, but it's an interesting read if you want to kind of follow some of the influential ideas of computational neuroscience. But right now, I'm just going to focus on two parts of this framing. The first is the K0 set. So what we can say about this, and essentially the ideas that were laid down in this type of in this framing, again as early as the 60s, is still basically what we use today in a canonical neural mass model. So the K0 set is a canonical isolated or an individual neural mass or neural population model. Now, the typical, well, the way that was structured by Freeman in the 60s and the 70s, and again that we still use today in the majority of these type of models, is we decompose or we describe the components of a single neural population in terms of two operations or two operators. They have these nifty names. We call them a pulse to wave operator and a wave to pulse operator. Now, pulse here is basically talking about firing rates, and wave is talking about membrane potential. And what these two describe is essentially the inputs and the outputs, or how the population responds to its inputs and how it generates outputs. So on the left-hand side, another term that's used, probably a better term actually, for the pulse to wave operator is the synaptic kernel or the synaptic impulse response kernel. So this graph here, we've got it's a function over time that's describing the temporal evolution of a postsynaptic membrane, population average postsynaptic membrane response to a unit input at T0. So we have a rapid response and then a slow kind of exponentially decaying decline there on the order of a few milliseconds. The second function that I have on the right-hand side here, so the wave to pulse operator, or we call the sigmoid response function, unlike the operator on the left-hand side, which is spread over time and is a function that evolves over time, or spread over time, the wave to pulse operator is considered to be instantaneous. And what this is doing is this is converting the membrane potential, population average membrane potential, into a firing rate. So on the x-axis, we're saying when the membrane potential is at a certain level, then go up, and the firing rate is going to be at a certain level, and that's the it's described by this squash or the sigmoid function. So unpacking these a little bit more, the mathematical description of this synaptic impulse response kernel, there's two equivalent descriptions that are used and that are useful to think about. One is the kernel or the integral representation, which is what I've got at the top here. And this is just showing you, so we're kind of integrating over time, and they're essentially sliding a window over the this function and multiplying it by the incoming inputs. And that's what a convolution is. A convolution is a kind of moving window multiplication. That's your kernel representation of this system. Now, there's also an equivalent differential equation representation of the same process. And this is just like a mathematical relationship between these two. We can derive, or we can convert one of them into the other. The convolution or the integral thing is a little bit easier to understand, which is why I've listed it here. But in practice, what we use is the differential equations. So we write down a second order system of differential equations and then kind of split it into a pair of coupled first order differential equations for your membrane voltage and the time derivative of the membrane potential or the voltage, and then the time derivative of that, which is the current, and it's given by this this system of coupled differential equations. So this is what we use when we're doing simulations, and this is what you see in papers that are using this type of neural mass model. The second point that's worth just unpacking a little bit in terms of the firing rate function is that this is a population average response. Okay, now what this results from is a average of a set of we call Heaviside or step functions that describe the population response to a single neuron's response given its membrane potential. So basically what this red line here is showing is that an individual neuron is receiving a membrane potential, and then when it exceeds a certain amount, then it fires. Okay, and that's your step. So the idea is that we have this step function describing the response, the firing rate function for individual neurons, but we have a distribution of the thresholds, or if you like, the location on the x-axis where the step occurs. And the reason for that is like random connectivity and just random dispersion in the incoming inputs that any given neuron is receiving at a given time. But what that results in when you have a randomly distributed or Gaussian distributed set of Heaviside functions, you want to take an average over that, is you get this population average sigmoid response function that we have here. And that was the justification of the formulation that came from Wilson Cowan in the 1970s. Great. So final point I want to make, and I'm wrapping up because we have another 5 minutes in this session, is the K2 set, which I mentioned before. So the K2 set is when we have a pair of interacting neural populations, one of which is excitatory and one of which is inhibitory, and they have this negative feedback. Okay, the negative feedback is because there's inhibitory cells that are driving and they inhibit. Now this schematic, or this level in the K-set hierarchy, this describes like 90% of all neural mass models. And this figure that I've got here is a really nice schematic from a really nice review by David Liley a few years ago that shows that, you know, there's a number of classic canonical models that are listed here, and they all vary in terms of the number of populations, number of excitatory and inhibitory populations, and the topology of the wiring between them. But they all fall into this K2 set, you know, hierarchical description, and they're also differentiated in this figure by the form of the postsynaptic response, which isn't so relevant here. But this kind of goes to show that this is okay, sorry. And then the final point to make here is that when we have this type of arrangement where we have our two populations that are coupled by a negative feedback, those are the conditions for the emergence of oscillations. You know, in physiological terms, it's inhibition. In more kind of systems level terms, it's negative feedback. And you basically can't have oscillations in a system if you don't have negative feedback of this kind. A canonical example, probably the most widely used example of this type of K2 set neural mass model is the Janssen-Ritt model. So I'm just giving you a sketch of it here. You also hear more about this type of model from Dimitris in a little bit. So we have two excitatory populations, pyramidal cells and spiny stellate cells, and an inhibitory interneuron that's you know, inhibiting the pyramidal cell population there. And the equations describing that are the ones that I listed in the slide previously. And this is an example. This is like the canonical architecture for mesoscale neural population models that are aiming to describe the generation of oscillations in a cortical column, or if you like, a cortical microcircuit in a minimal fashion, right? With essentially like as simple as we can get, but not simpler. That's really the type of motivation for this type of formulation. Okay, final point is, because we're talking about oscillations, is power spectra. So one of the advantages and really nice things about working with these type of relatively simple and relatively tractable neural population models is that under certain conditions and in certain, you know, with certain approximations and so on, we can obtain what's called an analytic power spectrum, or expression for an analytic power spectrum directly from the model equations. So what that means is what you kind of normally do, if you like, when you're simulating neural activity with computational models and differential equations, is you write out your model and then you kind of numerically integrate, you chug through time, you generate a time series, and then you can study the frequency content of that time series as you would do any empirical measurement. Okay, with a certain class of formulations and kind of modifications of neural mass models, we can actually skip the middleman and go straight to an analytic description of the power spectrum. And this is really powerful. I mean, it's really powerful in terms of giving intuition and understanding of how frequencies are generated, but also practically, it's just great to be able to not have simulations that take a million years. I mean, you can do a lot more. So that's a really important feature of the type of things that we're interested in with oscillations in neural mass models. Fantastic. So I gave you a whirlwind tour of typical neural mass model formulations and some of the things that we use in the context of studying oscillations. I'm going to thank you for your time, show you a lovely picture of Toronto, and then I'm going to rapidly take some questions. So we have Jeremy's talk coming up in a minute and a bit. I have two questions up here. So what is the difference between neural mass and neural field models? Yeah, we can do that. And then I had in the chat, but I think it's worth mentioning for confusion, that example showing us it is biological motivated around biophysical. Sure. So I can answer both of those. And I did actually say motivated, but I also said biophysical. Okay, so the first question here is, what are the differences between neural mass and neural field models? The answer is neural field models include space. So a neural mass model, like strictly speaking, it's what we call a point process, as in there's no explicit description of space. Now, obviously you can say I'm saying it's here and I have one here and here and here. And that's, you know, that's like an interpretation, but you're not explicitly encoding space into the model. Neural field models, they're fields. So a field is a spatially extended or spatially continuous system. So you use the same type of equations for the neural population activity with the kind of population response and input and output functions, but you explicitly encode space. And what that typically leads to is rather complicated integral equations or rather complicated partial differential equations. So that's what a neural field model is. It's like a neural mass model with a spatially contiguous representation of space. Thank you, Ronaldo. And then, so final question here is, it's kind of a statement, I guess, but I'll agree with it, is to describe Wilson-Cowan as biophysically motivated rather than biophysical. And yes, I agree. Oh, it's Francis. Hi, Francis. Yeah, so this is a, this is the type of thing I would also think of as a continuum, really. It's derived, right? Wilson-Cowan is derived from a description of spiking neuron populations, or roughly spiking neuron populations. So, I think it's probably splitting hairs to say, okay, this is biophysical, this isn't biophysical, but rather think of it more as a continuum. But that is certainly, I think Francis is accurately representing how those kind of discussions tend to go. And certainly someone who's modeling neurons with Hodgkin-Huxley equations, it's not unusual for those people to object to a Wilson-Cowan equation being described as biophysical. And there are technical and mathematical reasons for that too. Totally agree with that, Francis. Great. So on that note, I will draw this session to a close, and we can continue the discussion of some of these interesting questions about biophysical, physiological terminology and so on in our later chat sessions. And I will encourage everyone to go over to Jeremy's talk, which will be starting in a second. Thanks.